The Esprit BTK process for degradation and resorption was documented via a series of histological findings in experiments in preclinical models. And in this porcine model, you see the implantation at 6 months proceeding out to 4 years. And what we see there is largely what is described in our degradation curves and degradation data, which is you implant a square-looking strut. It forms a neointima around it that is fairly disorganized and is fairly, a reaction of the implantation to cover the struts. This is what we're looking for, to embed the struts in tissue so that the degradation process can go in a controlled way inside the tissue, along with mass resorption eventually. But degradation starts right after implantation, governed only by water and temperature. Temperature is well controlled in the body, so mostly water. Water is a very small molecule compared to the molecular weight of this polymer, so it permeates throughout the bulk of the polymer and degrades all of the strata at the same rate, but slowly. The process for total degradation or molecular weight degradation is 3 years. So the first few years, all molecules are too large to escape. They may be half of the original molecular weight, but they're too large to escape. So what you see is polymer-looking struts, square, transparent to light, so in OCT they will look like black boxes. They're highly reflective of sound, so they are somewhat visible in IVUS. The IVUS signal for how white and visible they are is actually correlatable to the molecular weight that we find at each time point. So the struts degrade slowly. At one point, enough molecular weight has been lost for some molecules to escape. They will migrate, they go into a free water environment outside the strut, and they degrade immediately and get resorbed. Lactic acid is used as energy in the body. It disappears fairly quickly from the region. The process continues, but in that process of creating a void, a very slow creation of a void from the escape of these molecules, is typically compensated with cells around by secreting matrix and or migrating into the spot to fill the void. So what you see sometime around 2 to 2.5 years is the gradual replacement of the translucent material with either translucent cell matrix or cells themselves, to the point that at 3 years, you no longer see struts. You see only either some composition and mixture of cells and matrix. Cells, of course, reflect lots of light, matrix does not, so you see a variable response to light in each one of these sites. At 4 years, everything is normalized. Smooth muscle cell has populated the surrounding areas and the strut areas. The vessel is fully functional. These cells are functional and contractile. There's an intima that has formed long ago. The vessel looks like it did probably when it was created. It is a very correct structure of a neointima, that it looks like the original intima, and a neomedia, which looks like the original media.